And let's talk first about their modes of nutrition. So we've talked about this already in here, the different modes of nutrition that you would find in all living things, but you'd find all of them in bacteria, or in prokaryotes. So the first thing that you need to know about modes of nutrition is how are they getting their energy, right? So an energy source. And I think when we talked about this last time, we related it to the second law of thermodynamics, and we said, look, if you have no energy source, everything gets more and more disordered, and it'll eventually break down into nothing, so you always have to have a constant energy source. The two possible energy sources are the sun, right, or light, so we would call those photo. Or you could also have the other possible energy source, which is eating other stuff. Okay, we call those chemo. So that's chemicals, consuming some sort of chemicals. Then you need the actual like living thing stuff, like organic compounds. How do you make organic compounds, right? So we call that the carbon source. How do they get their carbon? And there's two ways that you can get carbon. You can either take it out of the atmosphere, which we would call auto. Those things are going to undergo carbon fixation. Take CO2 out of the atmosphere in order to produce organic compounds. Or you can just eat other stuff that's already done that. So we call that hetero, right? So that's consuming organic material. And so then you can combine each of these things however you want. So you could have photoautotrophs, you could have photoheterotrophs, chemoautotrophs, or chemoheterotrophs, right? And they all work, although photoheterotrophs are pretty rare, where you'd be getting an energy source from light and consuming organic compounds. That's definitely the rarest combination, but all the other combinations definitely exist in fairly large numbers, okay? I also want to talk about oxygen requirements for these modes of nutrition. There are three types of oxygen requirements, or three different types, right? So you've got the first type, which is called obligate. And if you have an obligate anaerobe, those organisms are poisoned by oxygen. Okay, so obligate means it has to do it. Anaerobe means that there's no oxygen present. So in other words, this means there has to be no oxygen present. Okay, areas where there would be no oxygen present are areas that are maybe under mud or something like that. So these organisms might live in like a creek bed or something like that, or a swamp where there's not a lot of air that can get into them. Another place that you might find them is if you've ever, gotten a cut or something on a rusty nail, and they're like, oh, you should go get a tetanus shot. Right? The reason why you would go get a tetanus shot after cutting yourself with a rusty nail is because you are more likely to come into contact with the bacteria that causes tetanus on a rusty nail, because the bacteria that causes tetanus is an obligate anaerobe, meaning that it would be poisoned in the presence of oxygen. And rust, if you think about what rust is when oxygen is removed from the air and taken out and put into compound in a metal, right? So like iron oxide and stuff. That's taking the oxygen from the air and putting it in compound with the metal, and so it would then not be in the atmosphere to poison that species of bacteria. So you're more likely to find anaerobes in rusty areas, or obligate anaerobes in rusty areas, because there's not going to be as much oxygen present, because the oxygen is being removed by the rust, right, or by the process of rusting. Okay. You've also got obligate aerobes. Okay, these are organisms that must have O2. So they have no ability to go under any type of, or go through any type of metabolism that doesn't require O2, right? And then you've got the last type, which can do either one. They're called facultative. And you know that anaerobic respiration produces like 2 ATP per glucose, whereas aerobic respiration produces like 36 to 38 ATP. And so facultative aerobes are going to prefer oxygen metabolisms, but they will also be able to go through non-oxygen metabolisms. So they use O2 when available, but also have anaerobic metabolism. Okay. Where would we classify humans? You'd think obligate, right? But in fact, we're not. We're facultative. Because even though we can't survive forever on it, we do have anaerobic respiration. Our body can undergo lactic acid fermentation. And you guys have seen this all the time. If you go running or something like that, you know that the thing that actually makes you feel tired and makes you feel sore is when your body starts running out of the ability or doesn't have the ability to keep up with your oxygen consumption. Right? So if your muscles need more oxygen than your body can provide to them, then they start going through anaerobic respiration, which will start the buildup of lactic acid, which makes your muscles burn and makes your muscles feel tired. But as you get into greater cardiovascular health and you can transport oxygen around your body better, then that getting tired and feeling sore thing doesn't happen until you push yourself much harder. Right? That's because people who are in greater cardiovascular health don't go through anaerobic respiration as quickly as those people who are in less cardiovascular health because they can't transfer oxygen around their body as quickly. Okay. Now, will you eventually die if you only go through anaerobic respiration? Yes, but the reason why you die is because of the buildup of lactic acid. You don't die from lack of ATP. Your body can produce plenty of ATP going through anaerobic respiration, but as a byproduct, lactic acid is produced that will eventually decrease the pH of your blood and cause you to die from that, right? It's called acidosis. All right, let's talk about how bacteria move, or prokaryotes move. Prokaryotic movement is called motility, their ability to move, right? And almost all prokaryotic organisms move in the same way, and that is through a process that's called taxis, okay? Taxis means that they are moving towards or away from some specific stimulus. So movement towards, if it's towards, we call it positive taxis. Or away, if it's moving away, we call it a negative taxis from a stimulus. Okay, if I'm cold, if my body is cold, right, I have a very complicated process of moving myself to a warmer environment, right? So I'd be like, oh, I'm cold. I have these receptors in my body that tell me that my core body temperature is going down, right? Those receptors send a message to my brain that says, hey, we're cold. My brain says, oh, okay, I'm cold and I'm in a situation where I'm allowed to move, right? I don't have to remain here for any specific reason. And I know a place where it might be warmer, right? Then I send signals all over my body that say, okay, make my legs walk. And then I go to the place that's warmer and that warms me up, right? Bacteria don't do that. Bacteria are going through a much more simple process. Okay, so all they're doing is responding to a stimulus, and they can respond to a bunch of different stimuli. Things like photo, that's responding to light. Chemo, that's responding to chemicals of whatever type. Thermo would be responding to heat. And Rheo is responding to current, so the way that the water would be moving. Let's talk about how they do this. here's my bacteria. It's got a little flagella on it. Okay? So bacteria with a little flagella on it. This bacteria, let's say, has a positive phototaxis. So on the end of this bacteria, on the opposite side from the flagella, there are these photosensitive proteins that are going to catalyze some reaction whenever they come into contact with light. Right? So light hits them, it activates those photosensitive proteins. Right? That causes some chemicals to be released in the cell, which causes the flagella to move. Okay? So whenever it's facing light, that turns on the flagella. Right? Let's say that it slightly veers off course and it is going this way now. And now the light is not hitting those photosensors because it's faced sideways or whatever. Okay? And so it doesn't move at all. Its flagella turns off. Right? And when the flagella turns off, it doesn't have any directional movement. And what does it do then without directional movement? It just sort of floats around in its environment in 360 degrees. It just kind of hangs out. Okay? And eventually, through randomness, right, it will probably orient itself in this direction again. You know, it might orient itself this way, not do anything, this way, not do anything, this way, and this way. And eventually, it might point itself back towards the light. And when it points itself back towards the light, that's going to activate the flagella and cause it to swim towards the light. Right? Okay, how would we change this if we wanted, and that would be a positive phototaxis because it would move towards light. How would we change this if we wanted it to be a negative phototaxis? Put the proteins on the other end, right? So if the flagella is on the same side as the proteins, that means whenever the flagella is facing towards the light, it will swim away. Right? And then if it goes this way, it's not going to do anything, or this way it won't go anywhere. But when it goes like this, that activates the flagella and causes it to swim away. And this is the difference between a positive phototaxis and a negative phototaxis. You can imagine that an organism that is like photoautotrophic would really benefit from being from having a positive phototaxis, because that's going to make it move towards the light, which is going to mean that it's spending more time in the light, and that means that it's going to get more energy, and that means that it's going to be able to make more organic compounds. That means it's going to be able to reproduce more, and we know that organisms that reproduce more are more biologically fit and will pass on their genes to the next generation, and evolution, and yada 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 yada. Right? Okay, so let's talk about horizontal gene transfer. Horizontal gene transfer, what's up?
So it's only positive and
Yes. Yep, works for that one, works for that one, works for that one. Yep, that it works for all of them. Yep. Okay, horizontal gene transfer. So this is that weird thing that bacteria do where they just start expressing whatever DNA is in their cell. They have no control over what they express. They always just express any DNA that's in the cell. So bacteria can gain new genes during their lifetime and start expressing them. And there's three methods by which they do this. There's sort of like the generic term, which is called bacterial transformation. Bacterial transformation means that they're just going to take in random DNA. So let's say a bacteria dies, and when it dies, it releases its DNA out into the environment. Another bacteria might see that DNA and just take it in and start expressing it, right? So random DNA or RNA is taken in from the environment and expressed. Okay, so sometimes this works out well for the bacteria. Sometimes they pick up some random trait that's good and it allows them to, do something new or make an enzyme or something that they previously couldn't make. Sometimes it's bad and it kills the bacteria, right? But it's playing the numbers game here. Sometimes it's good, sometimes it's bad. If it's good, that means that it helped out the species as a whole, and it can then pass on those genes to the next generation. If it's bad, they die, and who cares, right? It's just one bacteria. There's millions of them, and they'll just keep reproducing. So it's no big deal. Okay. There's also what's called bacterial conjugation. And this is when bacteria share DNA in the form of plasmids. In the form of extra whoop, that's not long enough for that word. Extra chromosomal rings of DNA called plasmids. Okay, so the way this works, here's a bacteria, and it's got this little appendage on it that's called a sex pilus. Basically, it's like a spear, right? Like a thorn or something like that. And what it does is it goes and it just stabs other bacteria like this. Boom. And it pokes a hole in them. And when it pokes a hole in them, that plasma membrane is going to attempt to seal itself, and it seals itself by attaching to the other bacteria like that. Okay, so now there's no hole anymore, but now they are attached together. This area right here where they're attached together is called a cytoplasmic bridge. And the cytoplasmic bridge allows for DNA in the form of these plasmids to transfer across into the other bacteria, right? So they can share their plasmids back and forth. Okay, they can do this in between species, right? Or in the same species. And so this is a way where, let's say they got something really good by just taking in random DNA, right? Now they can share it with the rest of the colony by going through bacterial conjugation. Okay? The last one is the weirdest one. It's called bacterial, or sometimes it's called viral transduction. And this is a type of horizontal gene transfer where a virus accidentally transfers a gene from one bacteria to another. A virus transfers a gene from one bacteria to another. By accident. Okay, so in order to understand how transduction happens, you got to understand how viruses work. And we talked about that before, so we're going to hop back over here for a second, and we're going to look at these two cycles, the lytic cycle and the lysogenic cycle. Okay, so we said in the lysogenic cycle, what happens is that the virus is going to implant its DNA into the chromosome of the host cell. So this red part here represents viral DNA. Okay. Then, at a certain point, it will go through the lytic cycle. And in the lytic cycle, it's going to need to reproduce a bunch of that viral genome, right? How does it do that? Well, it's just going to say, hey, copy this section right here, okay? So let's say that this thing goes and it copies this viral section right here. Well, that'll just put that viral section into the next set of viruses, and then we'll release them out, and then they can go on to infect new things. But occasionally what happens, and what happens in viral transduction, is it messes up when it starts doing this copying here. And not only does it copy the viral genome, but it goes on and it copies part of the bacterial chromosome of its host cell. So now the viral genome isn't just the virus, it's the virus and then some gene from the bacteria, right? And now when this goes on and it releases all these things here, when they implant that DNA into the next host cell, guess what? They're not just implanting their own viral DNA, they're implanting the viral DNA plus this little extra bit here that got copied by accident, right? And this little extra bit on here might be something important. It might be some cool enzyme that the old host cell could make, but the new host cell wasn't able to make, and now it can because it has a little bit of DNA inserted into it from its previous host. Okay, so this is a lysogenic virus may implant DNA from its previous host into its new host, which can then leave that new host able to do something or make something that it previously couldn't. Okay? Let's see how much time we got left. 29, and we end at 41. Perfect. All right, let's talk about ecological impacts of prokaryotes. Carbon fixation, right? So because prokaryotes take CO2 from the atmosphere and convert them into organic compounds, they often act as the base of the food pyramid, right? So prokaryotes can use CO2 to produce organic compounds, allowing them to act as the base of the food pyramid, which is often called the biomass pyramid or the energy pyramid, but we'll talk about that later. So for now, we're just going to use the generic term of food pyramid. Okay. There are lots of types of bacteria that go through this carbon fixation process. We have both chemoautotrophs and photoautotrophs. Photoautotrophic bacteria are called cyanobacteria, right? And chemoautotrophic bacteria, there's a lot of different types of them, but they're generally going to use compounds like hydrogen sulfide for their energy source, but then fix carbon in order to make organic compounds. Let's talk about nitrogen fixation. Bacteria are the only organisms that can fix atmospheric N2 into NH3, okay? And this is so important, okay? Because all living things need nitrogen, every single one. We know that because nitrogen is in amino acids, and all living things have proteins, and therefore all living things have to have amino acids, all living things have to have nitrogen, right? If you don't have nitrogen, you can't live, and the only things that can take nitrogen out of the air, which by the way, N2 is like the most abundant gas in our atmosphere, but the only way we can use it is if bacteria first take N2 and convert it into NH3. And the only other way that N2 is converted into NH3 is through lightning strikes, right? So if we didn't have bacteria undergoing nitrogen fixation, then it would be really, really hard to have so many living things, right? Now, humans have figured out a way called the Haber process to make NH3 from atmospheric from N2 now. We just like pressurize it a bunch and heat it up. But anyways, that's like one of the things that allows us to make fertilizer and grow so much food. Otherwise, we would have to rely on only bacteria. But before we came up with the Haber process, the only way to get NH3, which is the necessary kind of nitrogen in order to give it to plants and living things, was to have bacteria do it. Okay? And then the last thing is decomposition. And there's this saying that goes that if there is an organic compound, there is a bacteria in the world that specializes in breaking down that organic compound, right? There's that many species of bacteria, and they're that unique, right? And so every bacteria is going to specialize in breaking down some compound, right? That's like what they do, okay? Decomposition means that there's some organic compounds that are too complex for other organisms to break down, but bacteria break them down and bring them back and allow them to re-enter the ecosystem by breaking them down into simpler parts. Okay, so bacteria break down complex organic compounds into simple ones that can be used by other organisms. Occasionally we will cultivate bacteria for the sole purpose of breaking down some organic compound that's produced as a byproduct of industry, and we call this process bioremediation. A good example of this is sewage treatment. We use bacteria for sewage treatment. We use bacteria because we know that bacteria can live on the sewage, and the byproducts that they produce from processing sewage are significantly less toxic than the sewage was in the first place, right? So this is using bacteria to process waste products produced by humans. So another good example of this is in the gold industry. So basically the way that you get gold out of a solution that has a whole bunch of other metals in it is to dissolve it using cyanide or cyanuric acid, right? And then after they do that and they get the gold out of that solution, they just sort of dump the cyanide, right? So they used to just be like, oh, well, this stuff is no good. It's contaminated now. And so they would just dump it into rivers and stuff. And as you can imagine, that's pretty bad for all the stuff that lived in the rivers. And so it would just kill off all the stuff in the rivers. And then the EPA came along and they were like, hey, you can't do that because you're killing everything. And so now they had to come up with a way of taking the cyanide out of all this waste that they were producing. And so they came up with a bacterial process, and they found a bacteria that, guess what? Its main mode of nutrition is digesting cyanide, right? It takes in cyanide and converts it into energy, right? So what do? They just have these giant plates, these rotating plates where they cultivate bacteria that break down cyanide, and then they just run the water through the plates, and the plates sort of circle around, and the bacteria digest away all the cyanide, and basically clean water comes out the other side, right? This is like one example out of millions of examples out there where we're using bacteria in order to sort of do our bidding or help us to break down some product that we don't want anymore. Questions on prokaryotes? Anything that we talked about today? Great, that's it.